In this module, we look at the equipment used for PCR reactions, and we will also see how long it takes for one step to complete, which is 3 steps we talked about: raising the temperature to separate the DNA strands, annealing the primers, and extending the primers. So let's look at that. The first initial machine which was used for PCR is depicted on the screen here. Then Kerry Mullis is the person who invented this procedure. He received a Nobel Prize for that. As I've said, it had a great impact on human health and also society. You can see this machine has 3 chambers. These basically 3 chambers had different temperatures, and initially when people were doing this, they were physically carrying one reaction tube from one chamber to next. And at that time, Taq polymerase was not available. The enzyme they were using would become inactive once it was exposed to higher temperature. So at each step, new enzyme had to be added. Now, we basically have a machine which can do these 3 steps in a single space. You put the reaction mixture in the PCR machine. It fluctuates the temperature in a regulated manner, how long we are going to maintain a specific temperature for. And now we have the Taq polymerase that is not inactivated when we heat the temperature to 95 degrees Celsius. I wanted to show you how this procedure was done initially. So here are the 3 cycles again: 95, 55, and 72. The reaction tube is placed in the machine. This machine has space, wells in which this tube can be placed, and then the machine is programmed what temperatures we want for how long of a duration. Then this reaction is allowed to carry out. Once the reaction is complete, say the 30 cycles have been completed, the tube is taken and for further analysis. How long does it take for each of these steps in a cycle? One cycle, as we know, is composed of 3 steps. First, we heat the temperature, heat our sample to denature or to separate the 2 DNA strands. That is done at 95 degrees roughly. From there, we lower the temperature to about 52 to 55 degrees or 65 degrees. And then again, we raise the temperature to about 72 degrees. This whole procedure takes roughly 5 minutes. In 5 minutes to complete one cycle. And we talked about in our previous module that after 30 cycles, we can amplify one single strand of DNA. We can make 1 billion copies from one single strand. So in this short duration, we can accomplish that much. Previously, the technique which was used would take more than a week to accomplish the same number. Here's the number. It took about a week to 10 days. We will talk about that technique also in one of our subsequent modules. Here in 30 cycles, meaning if one cycle is 5 minutes, 250 minutes or 2.5 hours, we can go from 1 copy of DNA to 1 billion copies of DNA. So this is basically the power of this technique, that we can start with a small sample which has very few DNA molecules, and we can amplify it. This is especially relevant, and it is very important. For example, when we are detecting low amounts of pathogen present in a patient sample. We will continue our discussion of PCR in the next module.